Good afternoon, everyone, and thank you for joining us for today's bite-size seminar focused on accelerating genomic diagnosis of disease. My name is Mara Jean Tilley, and I have the great privilege of heading up the Garvan Research Foundation, the fundraising and marketing arm of the Garvan Institute of Medical Research, and I'll be your MC today. Before we begin, I would like to pay my humble respects to the Gadigal people of the Eora Nation, the traditional owners of the lands and waters on which the Garvan Institute and St Vincent's are located. We recognise their continuing connection to land, waters and culture, and pay our respects to elders past, present and emerging. Our topic today is one that builds on decades of research, both here at Garvan and around the world. And it's focused on a very personal topic, our DNA. Almost every disease has a genetic component, and many of Garvan's scientists are involved in genomics research, studying the information contained in our genomes. What is a genome? It's the complete set of our DNA instructions. Garvan's vision is to use the information contained within our genome to impact and improve human health for better prediction, diagnosis, and for targeted treatments for disease. Today, we are thrilled to be joined by Dr. Ira Deveson and Dr. Kishore Kumar, who are collaborating on a groundbreaking new method to accelerate the genomic diagnosis of disease. First, we'll hear from Ira, who leads our Genomic Technologies team, and Ira is going to explain how his exciting new method is going to provide us with opportunities for new patient diagnoses. We're also going to hear from Dr. Kumar, who's a fabulous neurologist, clinician researcher, who's going to provide us with how he's translating this discovery into the clinic to find answers for patients, many of whom go through a protracted diagnostic odyssey waiting to find out the root cause of their disease. Because today is a speed date of our seminar series, super fast, there won't be an opportunity for live questions at the conclusion of the session. However, if you have a question that isn't answered through our presentations, please do submit it in the survey at the conclusion of the seminar, along with your name and email address, and we will get back to you on that. So, without further ado, I'm delighted to welcome Dr. Ira Deveson. Ira is a Senior Research Officer and Head of Garvan's Genomics Technologies team. He works as part of the Kinghorn Centre for Clinical Genomics, developing cutting-edge tools for research. Ira, it's great to have you with us. Over to you.
Hey, thanks for tuning in. My name is Ira Deveson. As Mara Jane said, I lead Garvan's Genomic Technologies group, and our goal is basically to develop and optimize new genomic technologies in order to improve the diagnosis of inherited diseases. And so we do a lot of DNA sequencing, and DNA sequencing has really been at the heart of all of the transformative genomics work that's been happening at the Garvan over the last decade or more. And all of that work has happened on these so-called shotgun DNA sequencing devices from this company, Illumina, who have basically enjoyed an unchallenged monopoly in the genomics domain forever. But right now there's a small but very interesting competitor in a startup company called Oxford Nanopore, who make devices that basically look like this, except that this image is not to scale. If it were, it would look much more like this. So these devices are very neat, sort of small handheld devices about as big as a stapler. And besides the size, nanopore sequencing has one very important advantage. So shotgun sequencing is called shotgun sequencing because the process involves smashing your DNA up into tiny little pieces before you begin. And these pieces are around 100 letters or bases in length. By contrast, nanopore sequencing devices sequence long native DNA molecules in continuous unfragmented pieces that are on the order of 10,000 to 100,000 letters or bases. And so when you analyze a given gene using shotgun sequencing, you first smash it up into these tiny little pieces, and then you use some sort of fancy algorithms to stitch these pieces back together. And this is kind of like doing a jigsaw puzzle until finally at the end, you have this continuous consensus sequence, which is your result. And that's all very well. In most cases, that's very doable. But consider this scenario instead, where you have a gene that contains what's called a short tandem repeat, or a sequence that basically repeats itself again and again, CAT, 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 and so on. Now, when you try to analyze this by shotgun sequencing, it basically can't be done, because when you shear this up into tiny pieces, they actually all look the same. And so it's like doing a jigsaw puzzle with tiny pieces that contain no discerning features. So there's no way you can stitch these together into that final product. And that's just an inherent limitation of shotgun sequencing that can't be solved with any smarter algorithms or anything else. Nanopore sequencing doesn't have that problem, because in a nanopore experiment, you just feed this long molecule into the machine and it reads straight through your repeat in one single continuous piece. There's no jigsaw puzzles required. And that might sound like a pretty niche example, but these sequences, short tandem repeats, are actually very common. They make up around 7% of the human genome, and they're also polymorphic, which means they differ in length between individuals. So one person may have 6 of these CAT repeats, another person might have 8 of these CAT repeats, and a third person has 11. But in addition to that natural diversity among healthy people, there are also certain repeats in the genome that become considerably expanded in certain individuals. And so instead of maybe 11 CAT repeats, you have 50 or 100. These things can get really, really large. And these highly expanded repeats actually act as disease-causing mutations. So it's quite a complicated story with lots of different genes being associated with different diseases. But for whatever reason, many of these diseases are neurological or neuromuscular diseases. And the list includes some very well-known examples, such as Huntington's, Fragile X syndrome, ALS. And those are, I guess, the big names. But by our count, there are more than 40 known neurological disorders that are caused by these repeat expansion mutations in more than 37 different genes. And these genes, as I explained, they can't be read by shotgun sequencing. And so while the rest of the genetics world has kind of moved into the modern age, repeat expansion disorders are still diagnosed using some pretty old school molecular biology techniques, such as the Southern blot, which was invented by this guy here, Edwin Southern, 25 years before I was born. And Southern blots can tell you how big a piece of DNA is, basically by migrating it through a sheet of electrified radioactive jelly. It's a great technique, but suffice to say that's relatively slow, it's imprecise, it's labor intensive, it involves radioactive chemicals. And what's more, you're just testing for a single gene. So it's one gene, one test. And that becomes a big problem if you have 37 genes that are underpinning more than 40 different disorders, all with complex overlapping neurological symptoms. So this is a pretty important unsolved problem in clinical genetics. And I think it's a very compelling use case for this nanopore sequencing technology that I mentioned. With that in mind, my team has recently developed a targeted nanopore sequencing assay that will simultaneously test every known repeat expansion gene in a single simple test. This is what the data looks like. Here I'm showing you the gene HTT, which is the gene that causes Huntington's disease. And at a specific position within this gene, there is a tandem repeat sequence of CAG, CAG, CAG that's repeated 10 or 20 times in healthy people. And I'm showing you the exact repeat sequences that were recorded using our nanopore method for a single patient who has 2 copies of this gene, one from their mother and one from their father. So the repeat is sort of anchored to a unique flanking sequence over to the left, and then you can sort of read the size of the repeat off the scale underneath. And in this case, the person has one normal or healthy copy of the gene, which is 18 motifs in size, and they have one expanded copy of the gene that has 64 consecutive CAG motifs. And this is well into what is considered the disease-causing range for this gene. And so unfortunately, this is a positive diagnosis for the condition. So that was just one patient, but here I'm showing you the repeats that we've measured using nanopore sequencing for 12 different individuals. And you can see that they all have that same CAG repeat, but there is a diverse range of different sizes among the group. And 5 of these individuals have very long repeats that stretch past that disease cutoff. And so these 5 individuals are all clinically affected by Huntington's disease, whereas the other 7 individuals were actually healthy. So in this case, our test is very clearly and correctly distinguishing healthy from affected individuals. So that's just a single gene, but keep in mind that we are simultaneously measuring 37 different genes in every single test, and these genes are associated with 40 different neurological diseases. We're now in the process of optimizing and validating this test across a wide range of different diseases, and we believe that ultimately this technology has the potential to streamline and improve the genetic diagnosis of neurological disease. So I'll leave it there and finish up by just thanking my team who are listed here and the growing list of clinical collaborators we're working with, including Dr. Kishore Kumar, a neurologist from Concord Hospital who's affiliated with Garvan, who we'll be hearing from next. Thanks very much for listening.
Thank you, Ira. You did very well on the speed date. Thank you very much. Kishore, we'll invite you to join us on the screen, please. So as mentioned, Kishore is a unique individual in that he's a practicing clinician and also a clinical researcher at Garvan within the Kinghorn Centre for Clinical Genomics. Really, we are so privileged to work with clinician scientists for the very reason that they are able to take the incredible discoveries made in the laboratory and including the work that Ira's described and help rapidly translate that into the clinic. It also makes the issues that patients are dealing with on a day-to-day basis very, very front of mind. So Kishore, thank you very much for joining us. Over to you.
Thank you, Mara, Jean, and Ira for that very kind introduction. I'm a neurologist at Concord Hospital and also visiting scientist at the Garvan, and I'm lucky enough to work in a genetic testing lab where we test for these repeat expansion disorders. So as Ira nicely explained, you can get this stutter in your genetic code, and once it reaches a certain level, it can cause disease. So once it crosses this threshold, it can cause a disease, which is often a neurological disorder. And they tend to occur in certain types of neurological disorders, such as those that affect your coordination, which is what we call ataxia, which affects your cerebellum, which is responsible for much of your coordination. They can also affect your muscle strength, causing weakness of muscle, a term we call myopathy. And then it can cause 2 very serious, progressive neurodegenerative disorders that result in early death, which is motor neuron disease and Huntington's disease. So these are very serious disorders and disabling and life-threatening disorders. And we know that many of them are due to repeat expansions. For example, the ataxias are due to these often expansions, especially when they're called spinocerebellar ataxia, or this common cause of ataxia called Friedreich's ataxia. And the myopathies can be due to expansions when they're part of the myotonic dystrophy group of disorders. And then we have motor neuron disease, which may be due to an expansion in this gene called C9orf72. And as Ira has already mentioned, Huntington's disease may be due to an expansion in this gene, HTT. So how do we test for these disorders currently? Well, it's very cumbersome, time-consuming, and expensive. And we have to test for these disorders each separately. And the 2 techniques that we use also in my lab at Concord Hospital, the lab that I work in, is the Southern blot technique, which is what Ira mentioned, and also repeat prime PCR. But of course, Ira's technique and this technique can offer a new approach to diagnosing these disorders because he can take a long read of the DNA, which spans this expansion and allows us to accurately detect these disorders. Also, you can test for many at one time, and it's actually cheap to test this way. Now, here's an example of repeat expansion disorder that's been recently identified, and it's called neuronal inclusion, intranuclear inclusion disease. And you can see here on this MRI scan, the patient has this bright signal here on the MRI scan. And previously, to make this diagnosis, you would need to do a biopsy, either of skin, in this case, a kidney biopsy, so an invasive test. Recently, we've found that this disorder is due to expansion in the gene Notch2-NLC. Now, this has just been described, and there's no way to test for this in Australia except using IRIS technique. And here, IRA has been able to show this patient with dystonia and tremor has an expansion in the Notch2-NLC gene. I'd like to show you a video of another disorder called CANVAS, cerebellar ataxia neuropathy, vestibular areflexia syndrome. This patient is unsteady. They require a cane to walk. And when you twitch their head from side to side, there's a delay before the eye corrects. If you see that little delay, this is a sign of CANVAS. And only recently, it's been discovered that CANVAS is due to an expansion in this gene, RFC1. And we have no way to test for it in Australia, and only one laboratory offering clinical testing in the world that I know of. But Ira was able to test for this disorder, and he's able to show the exact sequence of letters in the expansion, which I think nowhere else in the world can we get it to that degree of accuracy. So why is genetic diagnosis important? Because it gives patients an answer, and they are so happy to be told what this terrible disorder is that they've had for many years, to finally have an answer and to end that diagnostic odyssey. You can also give them accurate genetic counseling and give them advice on family planning. And in some cases, when the disease, for example, is dominant, you can give them advice on how to stop the disease being passed on to subsequent generations. And you avoid invasive tests, and you can perhaps enroll them in clinical trials. This technique falls into part of the broad range of genomic research at the Garvan, including machine learning techniques, where we can pull out a genetic diagnosis from the genomic data, research being led by Prof. Leslie Burnett. So this technique can, repeat expansions can cause a lot of neurological disorders. Traditional methods are very slow and inaccurate. This new technique promises to diagnose these accurately, effectively, and cheaply. And I would like to thank the team here, and especially Dr. Ira Deveson and my donor, Paul Ainsworth Family Foundation, and all the patients who've contributed to the research.
Thank you so much, Ira. You also were very good at keeping to time. Thank you so much for that. And so that means that I have the opportunity to ask you both some questions. So I'll invite Ira back to the panel, if I may. Kishore, you've discussed the fact that many patients find themselves on a diagnostic odyssey for a number of years, and Ira mentioned that as well. What is it like as a clinician scientist to be able to give your patients an actual diagnosis, a genomic diagnosis that makes sense of sometimes very complex and unusual symptoms that they've had for many years?
I think it's tremendously rewarding to be able to tell them what the answer is. That alone, even if you took out all the other advantages, that's terribly rewarding, and the patients are very grateful in my experience. So I think this is a really wonderful thing. But we can now do so many things. It often changes their management. We've had patients who've been treated, for example, with strong immune therapy drugs. We're able to stop their immune therapy drugs and focus on supportive management or treatments that will actually work. And they avoid often dangerous invasive tests like biopsies. And we hope one day that there'll be some drugs to treat these disorders. And the best way to do it is to get a genetic diagnosis first.
Fantastic. And one more for you, Kishore. I think we're always conscious of the fact that our clinician scientists are juggling their patients and very long clinical hours, and then juggling their science. How do you do it all?
Yes, it's hard. I think, yes, my wife's very understanding, I should say that, and we're working often like late at night. I think that's the way to do it, and your research should be incorporated into your clinical work as much as possible. And then that both helps your clinical work and your research. Yeah. Fantastic.
Well, hats off to you, and thank you to your lovely wife as well. Ira, so much has changed in genomics in the last 5 to 10 years. What do you think the future looks like for genomic technologies? You've shown us an image of of a sizable machine going down to something not much larger than a USB stick. What's next?
Yeah, so there's devices that I'm talking about, the nanopore sequencing devices, that are transformative in I guess 2 ways. One is the very long reads that I talk about, which open up whole new sections of the genome and whole new types of mutations that we've never been able to investigate before due to the limitations of shotgun sequencing. So that's going to lead to all sorts of new discoveries and potentially new treatments and new diagnostic methods. But at the same time, the size of the device and the fact that it doesn't require any sort of capital expenditure or a big fancy laboratory to support it means that we can actually potentially take these devices to the clinic and perform our analysis right there at the bedside, or maybe take these devices to remote and rural regions where it's patients haven't had access to the kinds of technologies that we take for granted here in Sydney. And so I think those 2 aspects of this technology make it very, very exciting and promising for the future.
Fantastic. And what motivates you to do your work, Ira? What made you get into science?
I just find it fascinating. I've always been in love with biology ever since I was a kid, reading books about dinosaurs and that sort of thing. And I just think genetics is the most fantastically interesting subject, because it can basically explain how life works. Be that a dinosaur or be that human disease, it's in everything, and that's why I love working at the Garvan Institute.
Fantastic. And how did you 2 find each other as collaborators? Kishore, perhaps I'll start with you.
I can't exactly remember, but we definitely the disorders that Ira's technique is effective at definitely my area of interest. So these expansions disorders are all the things that I'm interested in, and the things that we check for in our laboratory. And so there's a natural overlap between long read and the expansions and neurological disorders. And this is a great place to work. We all talk with each other closely, and Professor Sarah Kummerfeld, I think, helped with that interaction. So yeah, it's a great place with so much interesting genomic research going on.
Yeah, fantastic. And Ira, is he a good collaborator?
Yeah, Kishore's a great collaborator. He's a really, really good guy to have around, and looking forward to being able to work together in person a little more next year.
Yeah, exciting times ahead. Well, guys, look, cannot thank you enough. I think that's been truly, truly fascinating, and really appreciate the thought that you put into making it such a concise presentation. To our audience, thank you so much for joining us. I can see that there are many, many familiar faces in the room today. Our wonderful, long-standing supporters, our partners for the future, our corporate partners, our science partners, our consumer organisations. What we do would not be possible without your support and without the support of the community. We are forever grateful. As the guys have alluded to, we are hoping to do more activity in real life, on site and face to face. So whilst we'll absolutely be looking to continue having virtual activities that individuals can join from Sydney or indeed anywhere throughout the world, we will be very closely watching the situation with COVID-19 as we come into the new year and seeing what types of activities we might be able to safely return to doing on site. So please do keep an eye out for that. As mentioned earlier, there will be a survey that's going to pop up on your screen soon. Please do complete the survey. Your full and frank feedback is sincerely valued. We do try to improve everything that we do, and your guidance on that is most appreciated. If you do have any specific questions you'd like us to come back to you about, just pop it into the survey along with your name and email address. Medical research is more important now than ever. So I know many of you already support us in many, many ways, but if you were inspired by Ira and Kishore, please do consider making a donation to Garvan. We are most in your debt for the support that you give in order to fund our important medical research. Look forward to seeing you at the next virtual seminar. Check out the website for more information. Again, thank you, Kishore, thank you, Ira, and thanks to the team behind the scenes for putting this together, and wishing everyone a wonderful day. See you soon. Thanks so much.